Picture this! Researchers at MIT and SMART recently achieved something remarkable. They produced clinical doses of CAR T-cell therapy, a revolutionary cancer treatment in a microfluidic device about the size of a deck of cards. This breakthrough could transform how we manufacture life-saving therapies, making them more accessible to patients worldwide. Here's something that'll blow your mind. The microbioreactor used in this research is a perfusion-based, automated, closed system with the smallest footprint per dose, smallest culture volume, and seeding cell number, as well as the highest cell density and level of process control attainable. We're talking about producing the same quality of medicine with 100 times less space and 10 times fewer resources. Think about it. While traditional bioreactors can be the size of a small building, requiring specialized facilities with complex utilities and massive infrastructure, the microbioreactor vessels could fit in your laboratory pocket, and the controlling equipment could fit on a laboratory bench. Yet they're producing the same biologics, the same life-saving therapies, just faster, cheaper, and more efficiently. It's like comparing a mainframe computer from the 1960s to the smartphone, except this transformation is happening in real time in laboratories around the world. Welcome to Apprentice.io's Industry Insights. I'm Frank Maggiore, and today we're diving into the world of microbioreactors, the miniature machines that are revolutionizing how we make medicines, vaccines, and even sustainable chemicals. We'll be exploring how working small is leading to enormous breakthroughs in biotechnology. We'll unpack the science behind these remarkable devices. We'll discuss how these miniature marvels work, examining their sophisticated sensors, automation systems, and the engineering principles that make them possible. We'll meet the key players, like the Amber systems from Sartorius, which have become industry standards, and explore how companies are using them to compress development timelines from years to months. We'll also discover cutting-edge innovations that sound like science fiction, but are happening right now. Perfusion systems that run continuously for months, 3D printed reactor components customized for specific cell types, and robotic systems that can run hundreds of experiments without human intervention. And we'll see how cloud-based manufacturing execution systems are connecting it all together, creating a digital ecosystem that's transforming pharmaceutical manufacturing. Most importantly, we'll share real-world examples of how this technology is already saving lives and what it means for the future of medicine. From accelerating vaccine development to making personalized cancer therapies more affordable, microbioreactors are at the forefront of a healthcare revolution. So whether you're a scientist working in drug development, a patient or caregiver interested in the future of treatment, or just someone curious about how technology is transforming healthcare, this episode will change how you think about drug manufacturing. Let's dive in. Alright, let's start with the basics and build our understanding from the ground up. So what exactly are microbioreactors? Simply put, it's a miniaturized bioreactor, a vessel where we grow cells to produce medicines, vaccines, or other biological products. But here's where it gets interesting. While traditional bioreactors can hold thousands of liters, we're talking swimming pool-sized vessels. Microbioreactors typically work with volumes between 10 milliliters and 250 milliliters. They come in sizes ranging from about the size of a Tic Tac container to a coffee tumbler. To put this in perspective, production single-use bioreactors may be between 2,000 and 4,000 liters, where traditional large-scale stainless steel production bioreactors might be 20,000 liters. That's enough to fill about 100 bathtubs. A microbioreactor holds less than what you pour into your morning coffee, yet these tiny vessels can do almost everything their giant cousins can do, and in many ways, they do it better. But don't let the size fool you. These tiny powerhouses are packed with sophisticated technology that would have been unimaginable just a decade ago. They have integrated sensors for pH, dissolved oxygen, temperature, and biomass. Some systems now include advanced optical sensors that can measure cell viability, metabolite concentrations, and even specific protein production in real time. They can control feeding strategies with precision down to microliters per hour, adjust conditions in real time based on cell behavior, and collect data every few seconds. It's like having a full-scale production facility shrunk down to fit on your lab bench. The engineering behind this miniaturization is remarkable. Single-use sensor spots on microbioreactors allow for online monitoring of individual pH and dissolved oxygen levels in cultures during the process. Integrated pitch blade impellers, similar to larger traditionally stirred mammalian bioreactors, such as the Amber 250 and the Biostat SDR, provide effective mixing without damaging cells. This means the physics of mixing, mass transfer, and cell growth in these tiny vessels closely mimics what happens at the production scale. The real game changer? Parallel processing. This platform enables the selection of the best clones, media, and feeds under small-scale bioreactor conditions, ensuring a seamless scale-up. Instead of running one experiment at a time in a large bioreactor, you could run 24, 48, or even more experiments simultaneously. Let me paint you a picture of what this means in practice. Imagine you're trying to find the perfect recipe for a new medicine. You need to test different cell lines, various nutrient combinations, multiple temperature profiles, and countless other variables. With traditional methods, you test one variation at a time, with each experiment taking weeks or months. You might spend years just exploring the basics. With microbioreactors, you can test dozens of variations at once, compressing years of work into weeks. It's like having 48 scientists working in parallel instead of one working sequentially. Here's what makes them truly revolutionary from an economic and an environmental standpoint. They use about 10 times less raw materials and reagents compared to bench-scale bioreactors. In an industry where a single batch of monoclonal antibodies can cost millions of dollars to produce, and where specialized cell culture media can cost thousands of dollars per liter, that's not just an environmental win, it's an economic game changer. The small starting cell numbers required compared to existing larger automated manufacturing platforms means that smaller amounts of isolation beads, activation reagents, and lentiviral vectors are required per production run. The automation capabilities are another key differentiator that sets microbioreactors apart. Single-use bioreactors save setup time, reducing staff costs and turnaround time. Everything from sampling to feeding to pH adjustment can be automated. The liquid handling systems are so precise they can add nutrients drop by drop, maintaining exact conditions for optimal cell growth. One operator can manage 48 parallel experiments that would traditionally require an entire team of scientists and technicians. But perhaps the most important advantage, and one that's often overlooked, is data quality and reproducibility. Because conditions are so tightly controlled and monitored continuously, the data from microbioreactors is incredibly reliable and reproducible. Temperature variations are measured within tenths of a degree. pH is controlled to 2 decimal places, and oxygen levels are maintained within narrow ranges. This means when you scale up to production, you can be confident that what worked in the microbioreactor will work in the manufacturing plant. This predictability is worth its weight in gold in an industry where a failed production batch can cost millions of dollars. Now let's meet the stars of our show and understand how this market has evolved. In the world of microbioreactors, few names are as recognizable as the Amber family, which were originally developed by TAP Biosystems and then acquired by Sartorius Stedim Biotech. I was working for Sartorius at the time of the acquisition, and even then, it was seen as a brilliant strategic move that boosted our presence at pharmaceutical conferences almost immediately. These systems have become the gold standard in many pharmaceutical companies worldwide, and for good reason. The Amber 15 is truly the Swiss Army knife of microbioreactors. Amber 15 cell culture is a high-throughput automated bioreactor system for 24 or 48 parallel cultivations at the 10 to 15 milliliter microbioreactor scale. What's remarkable is how this tiny system mimics the conditions of large-scale bioreactors with extraordinary fidelity. It has the same impeller design scaled down proportionally, the same sparging system for delivering oxygen, even the same monitoring capabilities, just miniaturized. Let me describe these vessels in detail because they're engineering marvels. These are the little tic-tac sized single-use microbioreactors that have micro-impellers no bigger than a pencil eraser, yet somehow they create the same flow patterns as industrial scale impellers. They even have the optical single-use sensors for pH and dissolved oxygen integrated into them. Imagine sensors thinner than a human hair that can accurately measure critical parameters for weeks at a time. Major pharmaceutical companies are using these systems for clone selection, essentially finding the best cell lines to produce their drugs. Implemented in cell line development laboratories worldwide, Amber 15 cell culture makes experimentation more cost-effective by saving on labor, facility space, capital, media, and consumables. When you're developing a new biologic drug, you might need to screen thousands of different cell lines, each potentially worth billions in revenue if successful. The Amber 15 makes this process not just feasible, but routine. Moving up in scale, we have the Amber 250 systems, which represent the next evolution in microbioreactor technology. The Amber 250 High Throughput Generation 2 tackles these challenges with fully optimized, multi-parallel, automated scale-down bioreactors designed to replicate large-scale systems, reducing time, effort, and cost in process development. These systems bridge the critical gap between microscale screening and pilot-scale production. They can handle up to 24 to 48 vessels simultaneously, each with a working volume of 100 to 250 milliliters. During my time at Sartorius, I was on the team that helped develop the scale-up software with the mathematical modeling to scale up from the Amber 250 microbioreactor vessels to the larger Sartorius bioreactor vessels up to 2,000 liters. This was more than just a technical project. It was about creating an ecosystem. We wanted to make scale-up easier and keep our customers within our product family by making it technically superior and economically attractive for them to do so. It was a brilliant strategy since we dominated the microbioreactor space and could offer seamless scale-up. The software was developed using computational fluid dynamics, mass transfer correlations, and decades of bioprocessing knowledge to predict how a process would perform at different scales. This meant a customer could run experiments in an Amber 250 and have confidence that the process would work in a 2,000-liter production bioreactor. This kind of predictability is invaluable in pharmaceutical manufacturing. But Sartorius isn't the only player in town, and competition has driven incredible innovation. All of the large life science industry suppliers have developed their own models of microbioreactors, each with unique features and capabilities. Millipore, now Millipore Sigma, has their Mobius system. Eppendorf offers their BioFlow series. Danaher's Paul division has their Micro 24. Thermo Fisher has various offerings. Cytiva, formerly GE Healthcare Life Sciences, has their own systems, and Applicon has specialized solutions. Even smaller specialized companies like M2P Labs with their Bioreactor systems have carved out important niches. In 2023 and 2024, around 20 new 48 parallel microbioreactor systems were launched, incorporating improved automation and real-time monitoring to optimize bioprocessing workflows and reduce operational costs. This explosion of innovation shows how vital this technology has become to the entire industry. Each system has its own strengths. The Bioreactor system, for example, excels at very high throughput screening with its ability to use standard microtiter plates. The Mobius Breeze specializes in perfusion applications. Some systems are optimized for microbial fermentation and others for mammalian cell culture. This diversity means that researchers can choose the perfect tool for their specific application. The competition is driving rapid innovation across multiple fronts. We're seeing systems with better automation, more sophisticated sensors, easier integration with analytical instruments, and seamless connection to digital platforms. Machine learning algorithms are being built into the control systems, enabling predictive maintenance and process optimization. The market is growing rapidly too. The global market for microbioreactor systems was estimated to be worth around $135 million in 2023 and is forecasted to reach a size of around $277 million by 2030, with a compound annual growth rate of 10.7% during the forecast period of 2024 to 2030. Real-world adoption is accelerated at an unprecedented pace. Companies like Synthego and Bristol-Myers Squibb are using these systems to compress their development timelines dramatically. CDMOs, contract development and manufacturing organizations, are investing heavily because microbioreactors allow them to take on more projects with less infrastructure, making them more competitive and more responsive to their clients' needs. The return on investment is compelling and often surprises newcomers to the field. While a single Amber system might cost tens of thousands to hundreds of thousands of dollars, depending on the configuration, it can replace millions of dollars worth of traditional equipment and dramatically reduce operating costs. But more importantly, it can shave months or even years off development timelines. In the pharmaceutical industry, where a drug can generate billions of dollars in revenue and patent clocks are always ticking, time truly is money. Getting to market even 6 months faster can mean hundreds of millions of dollars in additional revenue. Hold on to your lab coats because this is where things get really exciting. The innovations happening in microbioreactor technology right now are straight out of science fiction, yet they're being implemented in laboratories today. Let's start with perfusion technology, which represents a fundamental shift in how we think about cell culture. Traditional bioreactors work in batches. You grow cells, harvest the product, clean, and start all over again. It's like baking bread, one loaf at a time. But perfusion systems continuously feed fresh nutrients while removing waste and product, like a flowing river that constantly renews itself. The Mobius Breeze Microbioreactor is a 2 milliliter automated single-use perfusion cell culture platform designed to support gentle, adaptable, and reproducible cell culture processes. Imagine achieving the same productivity with 100 times less volume. The engineering behind perfusion at microscale is fascinating. These systems use various cell retention devices, from microscale filters to acoustic wave separators, to keep cells in the reactor while allowing medium exchange. Some use alternating tangential flow, ATF, scaled down to microfluidic dimensions. Others employ innovative membrane technologies or centrifugal separation. The challenge is maintaining sterility and preventing clogging in channels sometimes no wider than a human hair. The numbers are staggering as well and worth emphasizing. With high T-cell expansion rates, similar total T-cell numbers could be attainable with a shorter culture period in the microbioreactor, between 7 to 8 days, comparable to gas-permeable culture plates of around 12 days, potentially shortening production times by 30 to 40%. That's not just an incremental improvement, it's a paradigm shift that can fundamentally change how we manufacture cell therapies. 3D printing is another revolutionizing technology for how we design and build these systems, enabling customization that was impossible just a few years ago. The perfusion microbioreactor was designed with the help of computer-aided design and fabricated using the 3D printing technologies, fused deposition modeling, FDM, and stereolithography, SLA. Researchers can now custom design impellers with specific blade angles for optimal mixing, flow channels with precise geometries for perfect fluid dynamics, and even entire reactor geometries optimized for specific cell types or processes. Let me give you a concrete example. A research team working with a particularly shear-sensitive stem cells needed an impeller that would provide adequate mixing without damaging the cells. Using computational fluid dynamics simulation and 3D printing, they designed and produced a custom impeller with curved blades and specific tip speeds within just 48 hours. Testing and iterating on this design using traditional manufacturing would have taken months and cost tens of thousands of dollars. With 3D printing, they did it for less than $100. Robotics integration is taking automation to the next level, creating what we might call autonomous bioprocessing. Modern microbioreactor systems aren't just automated, they're intelligent. They can make decisions about when to sample based on growth curves, how to adjust feeding strategies based on metabolite measurements, and even predict when problems might occur using pattern recognition. In 2024, we now have integration of artificial intelligence and machine learning algorithms within microbioreactor systems. These systems now incorporate predictive analytics to optimize cell culture conditions in real time, improving overall process control and reducing manual interventions. But here's what really excites me, the sensor technology revolution. We're talking about sensors so small and sophisticated, they can measure individual cell health parameters in real time. Single-use sensors make our disposable bags and single-use microbioreactors even more intelligent. They deliver real-time information about the progress of cell culture processes and help biopharmaceutical customers to understand their processes even better and further optimize them. The newest sensors go beyond traditional parameters. Raman spectroscopy sensors can measure glucose, lactate, antibody titer, and even glycosylation patterns in real time without sampling. Impedance-based sensors can track cell density and viability continuously. Some systems now incorporate miniaturized flow cytometry for real-time cell characterization. We can even have sensors that can detect viral contamination or mycoplasma within hours instead of days. Process Analytical Technologies, PAT, is no longer just for large-scale manufacturing. It's being democratized through microbioreactors. These systems can measure critical quality attributes in real time, adjusting conditions on the fly to ensure consistent product quality. It's like having a tiny quality control laboratory built into each reactor, constantly monitoring and optimizing the process. The integration capabilities are particularly impressive. Modern microbioreactors can connect to mass spectrometers for real-time metabolomics, to automated microscopes for morphology monitoring, and even to genome sequencers for tracking genetic stability. All this data flows into integrated systems that can make sense of the complexity and suggest process improvements. The convergence of all these technologies is creating possibilities we couldn't have imagined just a few years ago. Researchers at MIT recently demonstrated a microbioreactor system that can produce clinical doses of CAR T cells, personalized cancer therapies, in a device small enough to potentially be used at the point of care. This is the first time that a microbioreactor is used to produce autologous cell therapy products. This isn't just miniaturization, it's a reimagining of the entire paradigm of how and where we manufacture medicines. Looking forward, we're seeing development of even more advanced systems. Organ-on-a-chip technologies are being integrated with microbioreactors to create more physiologically relevant models. AI-driven systems will potentially design and run experiments autonomously. The future of bioprocessing is being written in these tiny vessels. Now you might be thinking, great, we have all of these tiny reactors generating massive amounts of data. So how do we manage it all? This is where the story gets even more interesting. Enter cloud-based manufacturing execution systems, or MES. And this is where companies like Apprentice.io are fundamentally changing the game. Think of an MES as the digital nervous system of your manufacturing operation. Our Tempo Manufacturing Cloud accelerates the end-to-end drug product lifecycle by connecting together distributed teams and sites, helping them to execute better on the shop floor and optimizing their operations through enterprise-wide visibility. It's the difference between having isolated islands of automation and having a truly connected, intelligent manufacturing ecosystem. Here's where this matters specifically for microbioreactors. When you're running 48 experiments in parallel, each is generating data every few seconds across dozens of parameters. You're going to quickly drown in all that information. We're talking about gigabytes of data per day from a single microbioreactor system. The Tempo Manufacturing Execution System platform doesn't just store this data, it makes it actionable. Real-time analytics can spot trends humans would miss, predict outcomes before they occur, and suggest optimizations across your experiments simultaneously. But it goes beyond just data management. It's about creating a truly integrated ecosystem. Tempo seamlessly integrates all of your systems and equipment so you can connect your systems and equipment with no code, middleware, or APIs. This means your microbioreactors can talk to your analytical instruments, which can talk to your quality systems, all in real time. When your microbioreactor detects a shift in pH, it can automatically trigger a sample to be analyzed by your HPLC system, with results flowing back to adjust the process, all without human intervention. The collaboration aspect is huge and often underappreciated. Imagine a scenario where your process development team in Boston is running microbioreactor experiments, while your manufacturing team in Ireland is preparing for scale-up, and your quality team in Singapore is reviewing the data, all working on the same platform, seeing the same real-time information. The Tempo Manufacturing Execution System platforms offers integration with mobile tablets and voice-controlled wearable headsets, and is a major step forward in integrating global teams to operate in real time. This global collaboration capability has become even more critical in our post-pandemic world. Teams can troubleshoot problems together even when they're continents apart. A process expert in Switzerland can guide a technician in India through a complex procedure, all seeing the same data in real time. This democratizes expertise and accelerates problem solving. Regulatory compliance, often the bane of pharmaceutical manufacturing, becomes much simpler with integrated MES. Every action is logged, every piece of data is traceable, and electronic batch records are generated automatically. Tempo's GXP SaaS platform is a cloud-native solution that adheres to good manufacturing practices for ensuring that products are manufactured safely and consistently, requiring features like audit trails, data integrity, and strict access controls to comply with regulatory standards. The integration with AI and machine learning is particularly exciting and represents the future of bioprocessing. The system can learn from every experiment, building a knowledge base that helps optimize future processes. It's like having a digital assistant that gets smarter with every batch you run, recognizing patterns across thousands of experiments that no human could detect. Let's talk about real impact, the stories of how microbioreactors are solving some of healthcare's biggest challenges and changing lives. First up, CAR T cell therapy. This revolutionary cancer treatment involves taking a patient's own immune cells, genetically engineering them to recognize and fight cancer, and putting them back. It's personalized medicine at its most sophisticated. The problem, the manufacturing process is expensive, time-consuming, and very manual, which has made it challenging to scale to meet growing patient demand. The traditional process requires specialized cleanroom facilities that cost millions of dollars to build and operate. Patients often have to wait weeks or even months for treatment, and costs can exceed $400,000 per patient per treatment. For many, this puts the treatment out of reach, both financially and logistically. Enter the microbioreactor solution from MIT and Smart. Human primary T cells can be activated, transduced, and expanded to high densities in a 2 milliliter automated closed system microfluidic chip to produce over 60 million CAR T cells from donors with lymphoma and over 200 million CAR T cells from healthy donors. A clinical dose of life-saving therapy from a device smaller than a smartphone. The implications are staggering and worth exploring. This translates to lower cost of goods manufactured and potentially to lower costs for patients. This should help make these therapies accessible to patients who couldn't afford them before and potentially enabling treatment in hospitals rather than specialized centers. In vaccine development, speed is everything, and microbioreactors are accelerating the race against disease. During the COVID-19 pandemic, we saw how critical rapid development and scale-up could be. Microbioreactors are now standard tools in vaccine development, allowing researchers to test hundreds of conditions simultaneously to find optimal production processes. What traditionally took years can now be done in months. But it's not just about speed, it's about accessibility. Microbioreactor technology could help enable distributed vaccine manufacturing, allowing countries to produce vaccines locally rather than depending on global supply chains. This could be game-changing for pandemic preparedness and response. The applications extend far beyond human health, though. In the bioindustrial space, companies are using microbioreactors to develop sustainable alternatives to petrochemicals. In the field of biotechnology, microbioreactors play a crucial role in synthetic biology applications. They allow for the precise manipulation of microbial metabolism, enabling the production of biofuels, bioplastics, and other sustainable chemicals. Consider the environmental impact. A company developing a new bioplastic can optimize their process using microbioreactors, using 90% less resources than traditional development methods. They can test hundreds of genetic modifications and process conditions to find the most efficient production method. This acceleration in green chemistry development could be crucial in addressing climate change. Food and beverage companies are getting in on the action too. Orders increased in food and consumer goods by 60%. In the life science industry and pharmaceuticals, as well as biomedical, that increase was 43%. From developing new fermentation processes for alternative proteins to optimizing the production of food ingredients, microbioreactors are helping create a more sustainable food system. The alternative protein revolution particularly benefits from microbioreactor technology. Companies developing cultured meat can optimize their cell lines and growth conditions much faster than ever before. Precision fermentation companies producing dairy proteins without cows can test thousands of conditions to maximize yields. This could be the key to feeding a growing global population sustainably. Even in personalized medicine beyond cancer, microbioreactors are enabling new possibilities. For rare diseases affecting only a few hundred patients worldwide, traditional drug development isn't economically viable. But with microbioreactors, companies can develop treatments using minimal resources, making orphan drug development feasible. Instead of using large-scale equipment for small batches, appropriately sized microbioreactors make personalized therapies economically viable. The environmental impact deserves special attention here as well. By using less water, fewer chemicals, and consuming less energy, microbioreactor-based processes have a dramatically smaller environmental footprint. A traditional bioprocess development program might use millions of liters of water and generate tons of waste. The same program using microbioreactors might use only thousands of liters of water and generate kilograms of waste. In an era where sustainability is crucial, this technology offers a path to greener biomanufacturing. As we wrap up our journey through the world of microbioreactors, let's zoom out and see the big picture. We've explored how these tiny vessels are revolutionizing drug development, from the flagship Amber systems that have become industry standards to cutting-edge perfusion technology that pushes the boundaries of what's possible. We've seen how cloud-based manufacturing execution system platforms create connected ecosystems, and how AI-powered digital twins are opening new frontiers in process development. The key takeaway? Microbioreactors aren't just smaller versions of traditional bioreactors. They're a fundamental reimagining of how we develop and manufacture biological medicines. They're faster, allowing parallel processing that compresses years into months. They're cheaper, using 10 times fewer resources. They're more sustainable, with dramatically reduced environmental footprints. And they're more flexible, enabling personalized medicines and distributed manufacturing. But here's what gives me the most hope. We're just at the beginning of this revolution. By 2025, artificial intelligence will drive 30% of new drug discoveries, cutting costs and accelerating personalized treatments. The convergence of microbioreactors with AI, digital twins, and cloud computing is creating possibilities that we can barely imagine today. Think about the patient stories that will be written because of this technology. Today, CAR T therapy requires specialized centers and costs hundreds of thousands of dollars. In the near future, with point-of-care microbioreactor systems, we could see personalized cancer therapies manufactured in regional hospitals at the point of use, making them accessible to patients everywhere. The person with a rare disease, affecting only a few hundred people worldwide, microbioreactor-based manufacturing could make their treatment economically viable for the first time. This technology isn't just about making drugs faster or cheaper. It's about democratizing access to cutting-edge medicine. It's about giving researchers the tools to tackle diseases we've never been able to treat before. It's about creating a more sustainable future where we can produce what we need without depleting our planet's resources. So, what can you do? If you're a researcher, explore how microbioreactors could help accelerate your work. Don't be intimidated by the technology. Today's systems are designed to be user-friendly, and the learning curve is worth it. If you're in the industry, consider how this technology could transform your processes. The return on investment calculations often surprise even the skeptics. If you're a patient or patient advocate, know that these innovations are working towards getting treatments to you faster and more affordably. Ask about clinical trials using therapies developed with microbioreactor technology. Advocate for distributed manufacturing that can bring production closer to patients. And if you're simply someone who cares about the future of healthcare, spread the word about these incredible advances. Public understanding and support drive continued innovation and investment. The future of medicine is being written right now, not in liters, but in milliliters, not in massive facilities, but on laboratory benches around the world. And not by a few large companies, but by a growing community of innovators who believe that smaller can indeed be better. The microbioreactor revolution isn't coming, it's already here, and it's changing everything, one tiny drop at a time. From cancer therapies to vaccines, from sustainable chemicals to alternative proteins. Thank you for joining me on this journey into the microscale world that's having an oversized impact on human health. In an industry where precision and compliance are paramount, Apprentice's Tempo platform offers a cutting-edge manufacturing execution system solution. It's cloud-based, mobile-first, operator-centric, with the most intuitive UI in the industry, making it perfect for today's dynamic pharmaceutical manufacturing environments and for the production of the world's most complex drug products. Whether you're working on groundbreaking cell and gene therapies, biologics, or small molecules, Tempo provides a fully validated MES system that's both powerful and easy to use. It streamlines your processes, ensures compliance, and adapts to your specific manufacturing needs. So get off of paper batch records and discover how Apprentice's Tempo Manufacturing Cloud can transform your operations by visiting us at apprentice.io. Attention industry professionals, revolutionize your manufacturing process with the Apprentice Tempo Manufacturing Cloud, a manufacturing execution system built to streamline your operations, boost efficiency, and ensure compliance all in one powerful platform. Experience the future of manufacturing with our free 30-day trial. Scan the QR code on your screen now to unlock a world of innovation. Don't just keep pace, set the tempo as an industry leader with Apprentice. That's all for Apprentice.io's Industry Insights. I'm Frank Maggiore. Thanks for listening. Be sure to like and subscribe. Hit the bell icon for notifications when new episodes drop. Please comment on if you have any additional topics you would like for me to cover in this space, and we'll see you next week for more Industry Insights.